Welcome everyone to our 8th Immunomodel webinar. Today we talk about CAR T-cell therapy, precision immunoengineering, modern oncology. And we will have 2 talks and a joint discussion after the talk. So please stay until the end. You can write questions in the chat and also ask them personally. My name is René Winkler. I'm working at the Josep Carreras Leukaemia Research Institute in Spain. And let me leave a few words regarding our Immunomodel cost action. This cost action aims to foster research and innovation in the field of preclinical immuno-oncology models, with the ultimate goal to advance the treatment of cancer patients by improving their outcomes and quality of life. And if you are not a member yet, you can still join because the cost action will still run next year. And you can visit our website, immuno-model.eu, and join and find all other information regarding this action and so on. We will come now to the first speaker of today. It will be Ciprian Tomuleasa. He is a Romanian physician-scientist specialized in hematology and oncology. He's a research professor at the MedFuture Research Center for Advanced Medicine within the University of Cluj, Napoca in Romania. He's also a senior consultant in hematology. And his research training were, among others, the Johns Hopkins Hospital in Baltimore and the MD Anderson Cancer Center in Houston. He has co-authored more than 125 scientific papers and was involved in many breakthroughs. For example, he was part of the team that performed the first Romanian successful haploidentical stem cell transplantation. He is also a lecturer at the hematology department at the University of Cluj, Napoca, and he serves as a general secretary for both the Hematology and Cellular Therapies Committee under the Romanian Health Ministry. Today he will talk about the design and preclinical assessment of CAR T-cells targeting ROR1 for mantle cell lymphoma. So Ciprian, please.
So thank you so much for the generous presentation. And next, in the next few slides, I'm going to present some of the work we are currently doing in Romania, in Cluj-Napoca. And I'm going to present our ongoing project on designing and testing in the preclinical setting a CAR T-cell that targets rare orphan receptor 1. And as a proof of concept, we chose to use the mantle cell lymphoma. So first, we have to move a little bit on background information about the tyrosine kinase, receptor tyrosine kinase, and why we chose ROR1. So beginning with, I don't know, half a century ago, a lot of progress has been made in translational research in hematology and oncology. In the last 5-6 years, more than 20 receptor tyrosine kinase classes have been described, and we have a lot of them. And theoretically, all of them and each individual of it can actually be a potential therapeutic target for especially for hematological malignancies, especially lymphoma. These receptor tyrosine kinases are especially important because their signaling plays an important role in cell physiology, and also determines the proliferation rate and the differentiation rate of both normal cells as well as malignant cells, with a special emphasis on malignant lymphomagenesis. Tyrosine kinase receptors actually play a role in the extracellular communication from the outside of the cells. So they take information from the outside of the cell and they move it and they pass it around to the nucleus, and thus they play a role in differentiation and in proliferation. Thus, receptor of tyrosine kinases are important in cancer, in tumor biology, because either they gain a functional amplification or their duplication might play a role in the cellular processes. So the dysregulation of receptor of tyrosine kinases actually influence cellular processes. And a lot of, from a tumor biology point of view, a lot of tumor cells exhibit an oncogenic addiction to RTKs. Thus, the tumor cell survival is important, and it's supported by TRK aberrant activation. And we have a lot of, throughout time, we have a lot of targeted therapies being developed and already used successfully in the clinic, like EGFR receptors, like PDA-GFR receptors, I don't know, ROS receptor, RET receptors, or many others that have been shown to play a very important role in cell survival, apoptosis, cell migration, proliferation, growth, and resistance to treatment. ROR1 is an important receptor of tyrosine kinase, receptor of orphan receptor 1 that is expressed highly during embryonal development, and it plays a very important role in organogenesis. And once the baby or the human tissue grows older and older, and once we advance in age, theoretically, their expression drops because it's directly connected with tissue development and differentiation. In malignancies, this ROR1 has been shown to play an important role in the differentiation, and it has been shown to be ever more increased in very undifferentiated malignant cells, like is the case of mantle cell lymphoma. So our question and our hypothesis was whether ROR1 can actually be targeted in oncology. And we did a screening for a lot of immortalized cell lines, both hematological malignancies, lymphomas, myelomas, leukemias, but also for solid malignancies. And we chose as a proof of concept initially to test it in mantle cell lymphoma. Why? Because mantle cell lymphoma is an aggressive and a pretty rare form of B-cell non-Hodgkin's lymphomas that originates from the outer ring of small lymphocytes surrounding the lymphatic node. We can either have an indolent mantle cell lymphoma or aggressive mantle cell lymphoma, but usually in the clinic, as hematologists, we deal mostly with aggressive mantle cell lymphomas. And the current standard of treatment for mantle cell lymphomas is usually rituximab, an anti-CD20 monoclonal antibody, plus intensive chemotherapy. Mantle cell lymphoma has a high incidence in the Western world, so especially Western Europe and the United States. And thus, the treatment is mainly based on chemotherapy plus rituximab or intensive chemotherapy, sometimes using also bendamustine or other targeted therapies. But the treatment outcome and the remission rates are they are often, but relapses often occurs also. And once a relapse appears, these diseases are treated either with a targeted therapy using ibrutinib, a Bruton kinase inhibitor, so an RTK again, and or with salvage chemotherapy followed by autologous stem cell transplantation. So thus, these patients, we have an unmet need for this subtype of malignant lymphoma. And I don't have to explain, only briefly, the concept of CAR T cells, so the chimeric antigen receptor T cells, in which from a patient, after leukapheresis is carried out, the cells are expanded, are modified ex vivo, and then expanded ex vivo before they can be re-injected following a line of chemotherapy. And afterwards, their effect will be sustained throughout time against various malignancies. And currently in Romania, we have approved CAR T cells for B-cell acute lymphoblastic leukemia and patients with diffuse large B-cell lymphoma. And our question was whether CAR T cells can be used to target ROR1 receptor in mantle cell lymphoma, as we have shown that mantle cell lymphoma has a high percentages of ROR1 expressing cells. So we designed the CAR T cells, and then we chose as a suitable target ROR1, and then we tested them in vitro, and then also on murine model. So the design included an ROR CAR plasmid, and then that were injected into immortalized line, so a T-cell leukemia line that was afterwards used to test it in vitro. Of course, before we can test it for mantle cell lymphoma, we did a large screening of RO1 receptors for all type of malignancies in our lab. So we tested it also for acute megakaryoblast leukemia, acute promyelocytic leukemia, myelomonocytic leukemia, but also some other aggressive lymphomas. And we have shown that this receptor is highly expressed in mantle cell lymphoma and Burkitt lymphoma. After we activated cells in vitro using Dynabeads, then we tested them in 101 percentages for lymphoma cells. And we have shown that after adding anti-ROR CAR T cell, the number of malignant cells started decreasing after 24 and 48 hours. Then our postdocs tested how the direct cellular implications of these CAR-based treatments, and we measured the LDH activity and also the release activity of TNF-alpha, interleukin-6, and interleukin-8 and 18, and shown that after the co-cultivation of mantle cell lymphoma cells and CAR T cells, this we had a decreased freedom of TNF-alpha and interleukin-6. And afterwards, and what we are currently working on is working for a preclinical model or testing it in a preclinical murine model of mantle cell lymphoma. So we just obtained the mantle cell lymphoma murine model, and next week we actually are going to test it again in a murine model. We didn't have it shown here, but maybe on our next talk we can have the results in which we test the RO1 CAR T cells for a murine model of CAR T cells. And the basic conclusion for our first test was shown that the CAR T cell were efficient, but the efficacy is limited due to the low activation rate. We still have to do some activation adjustments with cytobeads, and that ROR1 can actually be used as a therapeutical target for the treatment of mantle cell lymphoma. And as perspectives, our department plans to improve the CAR T cell efficacy by various activation steps. We are currently also working on inserting a ICAS9 suicide gene to remove the CAR T cells after the antitumoral effect has been shown, and also to transfer this technology to primary T cells. Thank you very much, and it's my pleasure to show some of the preliminary work and to discuss the results here.
Great. Thank you very much. We will discuss the results after the next talk.
Oh, sorry.
So we do a joint discussion, but feel free to post all your questions into the chat, or you can also let me know later if you want to ask the question personally, and we can make it happen. So
Okay.
with that, thank you very much, Ciprian, and we will move on to the next speaker. Jordi, can you already start your presentation, please?
Okay, yes. Can you see?
Yes. Can you? Yes, perfect. Full screen now. All right, great. And now it's a pleasure for me to introduce Jordi Minguillón. He did his studies in Barcelona, at the University of Barcelona. He has degrees in biology and biochemistry and a PhD in immunology from Pompeu Fabra University. And if I see correctly, then he moved to Madrid, where he stayed at the IdiPAZ CNIO Joint Pediatric Oncohematology Unit in late 2021 as a senior researcher led by Dr. Antonio Perez Martinez. And since February last year, he holds a permanent position at CIEMAT-IIS-FJD Joint Advanced Therapies Unit, also in Madrid. And he has co-authored 27 scientific publications, presented over 50 national and international conferences, registered 3 patents, received 2 orphan drug designations from the European Medicine Agency, And as a principal investigator, he secured 8 competitive grants, co-supervised 3 doctoral theses, and 2 or more are ongoing, and mentored numerous students. He's interested in both basic and translational research, and wants to bring the results to the clinic. His current lines of work include genomic and transcriptomic preclinical and translational research to treat rare cancers, and CAR T-cell therapies for pediatric solid and hematologic cancers. And today, he will present a talk on CAR T-cell therapies for pediatric cancers, right? So, Jordi, feel free to start.
Thank you, René, for this kind presentation, and also Barbara and IMMUNO model construction to invite me for giving this presentation. I divided this talk in these 3 parts. We'll make a short introduction on pediatric cancer. Then I will talk a bit about CAR T-cell therapy, although Ciprian also did an introduction, so I will go quickly in this part. And then I will talk about a bit about what we are doing in the IdiPAZ hospital in Madrid regarding our advanced cell therapy program, mainly a clinical research project that I'm involved in, and as well as with this translational research project on T-cell therapy. So we are, I'm a part of Antonio Pérez Martínez group here in IdiPAZ in Madrid. And our group is called the CRISP Unit of Advanced Therapies because the CRISP Foundation is supporting heavily us since the last 8 years. Thanks to them, we have a clean room facility with Prodigy closed systems to make GMP production of these therapies. And thanks to that, we started this CAR T-cell program, this advanced cell therapy program 5 years ago. And since then, we have treated over 40 patients with commercial CAR Ts and also with investigational CAR Ts. So regarding, we are working mainly on pediatric cancer, and just a few things about it. A good thing that we could say is that it is a rare disease. The prevalence is low, has a low frequency. It has also, we could say, a good overall survival. Over 80% of the patients survive to this disease in comparison with adult patients that have on average 70%. They can tolerate more doses of chemotherapy, so they have more chemotherapy tolerance. And also, thanks to that, they have an increased response through this chemotherapy and also radiotherapy. But on the other side, it's not so good, this survival. It's not 100%, because this means that we have a 20% mortality, and this is dramatic for being children and not elder people. Also because there are some subtypes of pediatric cancer, especially some kind of solid tumors such as DIPG, DMG, or ATRT, whose overall survival is very, very low. It's about 1 year or less. Also, not so good is this long-term toxicity that comes after the chemotherapy treatment. And again, not so good is this rare disease, because this means that it's not so interesting for the companies to invest in therapies and approach to increase the overall survival of these patients. So putting some numbers, in Spain we have more or less 1,500 cases per year of pediatric cancer. In Europe, we can see 35,000 more or less every year. And with this 80% survival means that on the other side, we have around 300 deaths in Spain, over 7,000 deaths in Europe every year for pediatric cancer. Regarding the types of cancers, the most prevalence is leukemia. It's about 30% of the total tumor. And if we account with lymphomas, that is more or less 10%. This means that the other part, 60%, are solid tumors. And as I said, the overall survival is about 80%. Some tumors have a high survival rate, such as lymphomas or acute lymphoblastic leukemias, but others, such as myeloid leukemias, brain tumors, or as I said, DIPG, etc., that it doesn't appear here, we have a very, very low survival rate. The typical therapy that the standard therapy that they are receiving is surgery, is chemotherapy and radiotherapy. And in the last years, immunotherapy approaches have arisen, and also targeted therapy is being used nowadays with some success. And now let's talk a bit about the acute lymphoblastic leukemia, that it's about 25%, 30% of all the pediatric tumors. As you can see in this graphic, we have the overall survival of the standard therapy, the standard chemotherapy that has been used in the last 60, 70 years. As you can see at the beginning, so in the 1960s, the overall survival was about 10%, was very, very low survival. And this was thanks to that 10 years before, some authors found that derivative of folic acid was active in producing aplasia on this on the blasts, on the leukemia blast of these patients. And thanks to that, we could reach this 10%, because before was much even lower. And in decade after decade, these protocols have been improving, and as you can see, we have currently about a 90% of overall survival in the long term for the last chemotherapy protocols. But as you can see, if you compare the total 16 therapy with the total 15, this survival hasn't improved. So we have like a plateau of about 91% of these patients surviving to this cancer. So what we do with the rest of the patients, no? Because again, these are rare patients, but this 10% is very dramatic. We have to treat them. So I like to present this video. That is a T-cell lymphocyte detecting, interacting with a tumor. And as you can see, when it detects that it's a strange thing, it kills the tumor. So we usually have always, I don't know exactly the rate, but as we age, we usually have some cells that lose the control of their division. They become tumor cells, and our immune system is able to detect them and to kill them, mainly by the interaction of receptor and ligands. And when the tumor can avoid this detection or inhibit the activation of this detection, then is when we detect the tumor. Then is when the cancer appears. So 30 years ago, more than 30 years ago, this author and his team, Zelikensia, invented an approach to try to kill again, or to see again this or to avoid this evasion system that the tumor has by combining an antibody domain with the activation domain in the cytoplasmic of the T-cell receptor complex. That's the chimeric antigen receptor approach. This was a first generation approach because it had only one domain, one activation domain, and it had some success, some activity, but then we had more generations, more complications on this system. And as you can see here, we have a lot of domains that we can configure. And I'm not going to here in detail. And as I said, the first generation that was discovered was with an activating domain, cytoplasmic activating domain of the CD3 zeta. And then the second generation included one domain that was a co-stimulatory domain of CD28 or 4-1BB. After that, there are more generations that they include some other domains, such as to put 2 co-stimulatory domains, or to include in the system the release of cytokines or some factors that can improve the efficacy to reduce the exhaustion of these CAR T cells. So the first clinical evidence that this CAR T worked started more than 15 years ago, and as you can see, for leukemia, we had some papers published in 2011, and for pediatric cancer, the first patients were published in 2014. So this is Emily Whitehead, that is the first children that was treated with pediatric acute lymphoblastic leukemia with CAR T, with a CAR T19, that is a CAR T to detect the CD19 antigen. And as you can see, Stephen Grab here treating this patient for the first time. And this was the first case of a big success, because now, 13 years later, the patient is still alive and is considered cured. And this is very, very important, because for this therapy, it means that you can reach, in a good proportion of patients, stable remissions, stable cures. After these papers, after this 2014 paper, it was published some years later, the first report of the drug, considered a drug produced by Novartis, that is Hatieganu, or the commercial name that is Immunoengineering. And they showed like a 50% of overall survival. And as you can see, this was in 2018, and then the next years, this therapy was approved by EMA and by FDA, and some years later, other therapies were approved for the same target, for CD19, for leukemia and for lymphoma. And last year it was approved for 5. And later, in 2021 and 2022, we got also approved a CAR T cell therapy for the target BCMA to treat multiple myeloma. So nowadays we have 7 approved therapies to treat leukemia, lymphoma, and multiple myeloma. If we make a picture of what is going on in Europe, you can see since there are reports on this registry of the European Bone Marrow Transplantation team, they started to collect data in 2019, and after 4 or 5 years, they showed that they have treated in Europe over 10,000 patients, mainly lymphomas. And if you see that we have more or less 40, 50, 60% of overall survival of these patients that before all death died because they didn't have more options. This means that over 5,000, 6,000 patients are alive thanks to this CAR T cell therapy. If we see by country, we see that Germany is the first one, and in Spain we are in a good position. On almost, I think now about 1,000, 2,000 patients were treated in a cumulative way. So again, if we go to the standard therapy, we have this 90% plateau, and if we treat this 10% of patients that relapse, we use this Kymriah, this Hatieganu therapy from Novartis. The last report showed that had about 60% overall survival in the long term. So we can reach now, in this acute lymphoblastic leukemia, we can reach 96, 97% of overall survival. So this is good for acute lymphoblastic leukemia, that is of the B cell. That is one subtype, the most prevalent subtype of the leukemia, but there are others, such as acute myeloid leukemia or T-cell acute lymphoblastic leukemia. What happens with these other patients? In Europe, there are few options. Now, some clinical trials have been published in the last 2-3 years, we have now some options for this patient. This is a quite interesting case that was published in 2022, Eliza, that was treated with a CAR-T at the 37 marker, but it was important because it was also the first patient that was treated with a CAR-T cell with genome editing to avoid T-cell fratricide, because CAR-T cell for TOL is not the same as with BOL, it has some more difficulties, and also it was an allogeneic therapy, it was not an autologous such as with family Whitehead. This was done by Vasinka's team and Robert Chisa's team, and from UK, and in 6 months ago, no, September of the last year, the team of Franco Locatelli and Dario Campana from Italy and Singapore, they also published a clinical trial showing very nice results with another approach that also used CD7 as a target for CAR-T cell therapy, also fratricide resistant. So I put these examples because if we see in what positions we are, in Europe, we are not bad, but we are not also first on this game. China is the first, who is doing a lot of research, clinical research and translational research. USA is the second, and we are in a third position. This is not bad, but it's not the best, because these patients, for instance, this Elisa, if it was born on Spain, we couldn't have treated her. So we still have to do a lot of work. So here's where our CAR-T cell program wants to make an effort to reach this patient as soon as possible. One thing that in the hospital we are doing is to combine. The first thing that we do is to combine this CAR-T cell therapy with NGS sequencing. Every patient that arrives to the hospital is sequenced with a built NGS panel that we call Mood4Child to detect over 600 specific genes that have high prevalence mutations on children. And thanks to that, we can identify high-risk patients from this, for instance, from this leukemia, acute lymphoblastic leukemia patients, such as PAX5 or TP53, that when these mutations arise, the relapse after a CAR-T is very high. The proportion of relapse is very high. So if you detect a mutation on this patient, what we are doing now is to give the patient the Kymriah, the commercial therapy, but after that, we are doing a hematopoietic stem cell transplantation, because for sure, with a high probability, this patient will have a relapse. And with this hematopoietic stem transplantation, an haploidentical stem cell transplantation, we have an improved rate of survival and reduced rate of relapse. About 10-20% more than is reported with Kymriah alone. So this would be a first approach. But what we can do after that, because we still have patients that relapse. And in the world, some hospitals and researchers are trying to do multi-targeting approaches, dual targeting or 3 targets at the same time to detect not only CD19, but also others such as CD22 or CD20. For pediatric leukemia, this CD19 and CD22 is the most studied, and we have some different approaches to do that. We have this co-administration approach, that this means that we have 2 CARs, 2 products, and then we infuse these products at the same time. We have a sequential approach, that it means I infuse a CD19 to the patient, and then 1 month later, I infuse a CD22. We can do co-translation. This means that we have 2 vectors, such as in co-administration, but only we do 1 production. And then we have these CAR-Ts have mixed proportions. Some express CD22 CAR-Ts, other CD19, other both. And then we have 2 options with 1 vector. One is called bicistronic, that this means that the vector has two proteins, and we are also doing one product in fusion, one CAR-T, but this vector expresses in the membrane proteins that detect CD19 and CD22 independently. And finally, we have this tandem CAR approach, that it means that we have a one vector, only one product, as well as in bicistronic, but with one protein, because in the extracellular domain, we have two domains that detect the two proteins. Why I explain that? Because these 5 different approaches have been published in clinical trials, the majority in phase 1 and one in phase 2. They all showed good overall survival response, but none of them in Europe. So we had a lack of research on this, of clinical research on this. They were published on UK, on China, and the United States. So here stands a collaboration that we started with Miltenyi Biotech Company about 5 years ago in 2020, where they developed preclinical in their facilities this tandem CAR against CD19, CD22. They do the preclinical validation, and they gave us the virus so we could validate them in our clean room for do a production, a CAR product with in GMP conditions to be ready to use them in patients. So once we validated them, as early as in 2020, we started a compassionate use program to treat these patients on compassionate use, and also we started the paperwork to obtain the approval from the Spanish regulatory agency of a phase 1 trial. That was a bit difficult to obtain. So in the last 5 years, we could treat 11 patients by compassionate use. I show here the 10 patients because it's the publications that we submitted. And as you can see, from these 10 patients, one of them that was the first is the one that survived the most, and he's still alive, more than 4 years now. And we had 4 patients that died for different reasons. 2 of them, E2 and P3, they didn't express CD19, and they expressed at low quantities of CD22. So the relapse probably was to antigen escape mechanism. The patient 4 also had low expression of CD22. The patient 7 had a problem with infections, but this patient has Down syndrome, so these patients are very difficult to treat, and they have more toxicity for any therapy that you give them. But the good thing is that for these 10 patients nowadays, we have 6 of them still alive, and all of them were on relapse or refractory to other therapies, other chemotherapy, other CD19 CAR-Ts, or also refractory to previous stem cell transplantation. So if we see this Kaplan-Meier survival curve, you can see that we reach about in about 1.5 years an overall survival of 70%. And thanks to the paperwork that we started 4 or 5 years ago, we obtained the approval from the regulatory agencies last November, and we are still doing the paperwork to open the trial and hope to be open soon. So these 4 the clinical aspects of this CAR-T that it was in collaboration with this company, with Miltenyi Biotech. So we are also doing research with other diseases, with other CAR-Ts, and in all these CAR-T cell discovery programs, you identify a marker, as Ciprian explained that he identified this ROR1 for this mantle cell lymphoma. And in this case, when you identify this marker, you have to define if you want to detect this marker with an antibody, to make an antibody-based CAR T, or with the ligand-based. There are some examples with only the ligand, such as with NTJ2D or others. If you want to do it with an antibody, or you use already published antibody, or you have to make your own antibody to identify then the variable region of the antibody, that is the single chain fragment variable, to clone it in a CAR vector, in a plasmid, and then to validate it in vitro and in vivo. After that, once you do the preclinical validation, then you have to validate in a clean room that your product can reach the good medicinal practices rules. And after that, you can treat patients. So that's what we did with Miltenyi. But as I said, Miltenyi gave us the virus. So they did all this step until the preclinical, until vivo. And we have other CAR Ts ongoing in this pipeline. Two of them for T-cell acute lymphoblastic leukemia, others for also for TOL, for acute myeloid leukemia, and we have another one for solid tumors to treat all the solid tumors that with this target we now know that all these tumors express this marker. So I don't know if I'm good on time. René? I'm okay with the time?
Yes, you are okay. Like you can still have 5 more minutes, and then we come to the discussion round. I can see that Ciprian is already active in the chat, but we will have enough. We have enough time. Yes.
Okay, so I wanted to see if I have time. I explain as an example how we started a CAR T-cell program from the scratch, so from the identification of a new antibody. This solid tumor marker that we are developing for treatment of these solid tumors. So for this approach, for this CAR T, we follow these 4 steps. First, antibody production, then antibody characterization to see if it's good, etc. Now I will explain. Then to identify the single chain fragment variable, and then to clone it in a vector, in a plasmid, and then to validate that this vector works in lymphocytes. And this is where we have reached now. So after that, you have to validate in vivo, etc. And as I said, in vivo, then GMP, etc. So to obtain this antibody, we started this 2 years ago. We immunized mice with cell lines that expressed mouse cell lines that expressed this solid tumor marker, human solid tumor marker, in the membrane surface, and we injected them. After some weeks or months, we made a second immunization, this time with the recombinant protein, with the human recombinant protein. And after that, when we detected by ELISA that the blood of the mice were positive for antibodies to detect this marker, then we remove the spleen of the mice and we fused them with myeloma cell line to make these hybridomas, what we call fusion, and we make these hybridomas that will be will divide forever, will be like a cell lines, and each clone we make limited dilution to isolate monoclonal clones and then to characterize them. So for antibody characterization, this was done with the help of the CNIO antibody facility. And after that, this facility wanted to characterize more the antibodies. We obtained 15 clones, 15 positive clones, monoclonal clones, all of them. And they characterized them by immunohistochemistry, by ELISA, and we in the lab with PhD students, we validated by cell cytometry. As you can see, we obtain X cells that are deficient for this, are knockout for this solid tumor marker. And as you can see, all of the clones were negative. They didn't detect the protein as they were knockout. And when we used the wild type X cells, they were positive. As you can see with the controls, and then when we use an antibody, you have an antibody against solid tumor, has a very big, intensive expression, and some of the clones, all of them I don't show here, but all of them expressed, were active to detect this marker, but I show here the clones that express the most intensity. And then we selected these 3 clones. So for us, we finished with the antibody selection. We also characterized the biochemical affinity of these clones, of these antibodies. This is an in vitro characterization. You use the antibody and the recombinant protein that we immunize with the mice, and we use this bioligand interferometry system, and they show you when you treat when you use this assay, you obtain a constant affinity of this antibody for this protein. And through these 3 clones, we had 2 clones, 1 and 3, that had a good affinity constant, that is about 2 nanomolar, and this is good. And we had 1 that was not so, 140 nanomolar. But we still wanted to see if this worked on T lymphocytes, etc., because it's important that you have a good affinity. But also it's important that when you detect the ligand, in this case, our ligand is a long ligand, and it's not the same if the antibody detects on the top of the ligand or in the base of the membrane zone. That this means that when they interact, the immunological synapse may be better or not. So you have like some nanometers of distance that are optimal to do this immunological synapse. So it's not all that you have a good antibody of good affinity, but the region that is detecting this marker. So we identified the variable region, and then we send all the clones to this company, Apterra Bio, to sequence these clones. We wanted to sequence all of them because this target has been already published, and there are other antibodies commercial antibodies for this target. So we wanted to sequence all of them just in case the most important ones is the 3 that are the best ones. Their sequence was already published, because we wanted the intellectual property to move to a good CAR T-cell program development. These are the sequence that we obtained. All of them were different. 2 of the clones were the same, that is in green. So this means that from these 15, we had 14. And 1 of them was an IgM, that this is not a good antibody. So finally, we had 13. And from these 13, we selected, as I said, these 3. From this variable region, we cloned this single chain fragment variable to this second generation CAR T design. And as you can see, we have this design, the single peptide with a flat epitope to be detected with commercial antibodies, with the SCFB, a hinge to make mobility to this SCFB, transmembrane, 4-1BB costimulatory domain, and CD3 zeta activating domain. This was done with the help of the GenScript company. And finally, for the CAR validation, so we have this data from the last 2 months. So this is a program that started 2 years ago, and finally we are having good data now. And once we have the plasmids, we have to be sure that this protein can express in mammalian cells. And as you can see, we did a transient transfection in HEX, and all of them expressed these CARs. We produced the virus. This is a lentiviral vector. We produced virus, and we checked that this virus could infect mammalian cells. And as you can see, we could obtain that better with the clone 3, not so good with the clone 2, and very low with the clone 1. We still don't know, but we are still doing that. And finally, with this virus, we transduce them with activated T cells. And as you can see, we have the clone 3, a good proportion of positive cells, and lower with the clone 2 as before, as with the virus, and not so good with the one. And now we are doing cytotoxicity experiments with this, and also repeating the virus production because the first clone was not so. So this was an example of a CAR-T cell development from the very beginning. And as concluding remarks for all the talk, is that with our CAR-T cell program and with the help of the approved therapies, we are improving the survival of pediatric patients that have hematological tumors, especially for this CAR dual, the B-cell acute lymphoblastic leukemia, that we are, if we add this identification of specific mutations, we are using the commercial CAR-T, and we add this tandem CAR, we are reaching now over 98% of overall survival in the long term with these patients. Now, the last patients with the tandem were compassionate use program, and now we have to validate this in the phase 1. So let's see what happens in the following years. We also started preclinical and clinical development to treat other hematological tumors such as T-cell acute lymphoblastic leukemia, myeloid leukemia, and other more rare. And also we are moving on for other targets to treat patients with solid tumors. And I wanted to show here like a bet, because some people can think or can ask, when is this going to happen? As I said, the phase 1 is approved for this tandem CD19/CD22, and the recruiting phase usually goes very fast. So my bet is that in the next year we will have all the patients treated, and for 1 year follow-up, then I think we could have results for the 2027, in 2 years more or less. For the CAR-Ts that we are preparing to treat the non-B leukemias, my bet is to start a clinical trial on 2027. We have validated cytotoxicity in vitro, and we are moving to in vivo this year, and then the GMP validation next year, 2027. So my bet is this 2027, 2030 to do this clinical trial. And for solid tumors, I think more or less the same, 2027, 2030. So that's all. I wish to thank all the team. This is a multidisciplinary team of PhDs, students, postdocs, clinicians, pharmaceutics, etc. Thanks to all.
Great. Well, thank you very much, Jordi. Also, thanks again, Ciprian. We will now come to the discussion round. So you can see that there's already a few conversations going on in the chat. We will quickly go through them, but you can still post all your questions there or raise your hand and we can enable you to speak. Okay. And the first questions were regarding Ciprian's talk. Yeah. I just will to recapitulate it if people missed the comments in the chat. So Ciprian, you commented that the antibody was raised against the extracellular domain of ROR1.
Is it yours, Ciprian, the antibody?
So what, excuse me?
Is it yours, the antibody?
No, it's
produced it?
It's from purchased, the antibody. It's from, yeah.
And you commented that you think it's mediated by interferon gamma produced by the your CAR cells. Yeah, they affected this killing. Yes. And we had another question, and that was if you checked other mantle cell lymphoma cell lines for ROR1 expression and how it looks with primary mantle cell lymphoma cells.
So we are doing now the stainings in the pathology department, like from a paraffin embedded tissue of mantle cell lymphoma to do immunohistochemistry classic. I think it's better than on using cell lines.
So do you see also an upregulation in the expression of the gene, or is it just like a higher surface expression that you can detect?
I think it's maybe a higher surface expression, but I'm not sure.
And just out of curiosity, I wanted to know, why do you think it is specific for mantle cell lymphoma? Do you think it has something to do with the cycling D1 translocation, that it can promote the
possible, it's possible, but it's not in hematology. We tested on all cell lines so that we have, but in mantle cell lymphoma, it's more than 90%. It's also expressed in Burkitt lymphoma, around 60%, in B-cell acute lymphoblastic leukemia, 30%, which has a lower expression. So it's not specific. But for mantle cell, it's higher expression than in comparison to other ones. And it's also highly expressed in melanoma, lung cancer, and triple negative breast cancer. More than 90%. And around 60-70% in primary hepatobiliary cancers, like cholangiocarcinoma and hepatocellular carcinoma.
Well, thank you. And please submit your questions if you have any, or unmute yourself and we can discuss everything. We still have a few minutes left to do that. I was curious because ROR1 is an orphan receptor, yes? Was the ligand identified by now?
No.
No. So it's not known what binds the receptor. Do you have any questions for each other, Jordi and Ciprian?
My question was this: the antibody, no, if they produced it, where did they get? No?
Well, we will wait a few more minutes, but if there are no more questions, then we will come to an end of the session. Jordi, you had a really nice overview of the current state.
Congratulations, very good, very good. It was a pleasure to listen to you.
Thank you.
Just out of curiosity, they are all second or third line treatments?
For Kindraya is second line, I think. I don't know if they are moving to first line, but it's an interesting point because it's not innocuous, no? It has some adverse effects, but these effects are not genotoxic. As you know, if you treat with chemotherapy, no, genotoxic chemotherapy, a child of 5 years, you have a good probability to have a second tumor 20 years later. No? So in this case, as we are seeing this very nice results with CAR T-cell therapy, for me it would be good to try with not second line but first line, no, as a first option, just because of that, because you are having adverse effects on the first month, no? Cytokine release syndrome, ICANS, etc. But there are some reports that say that very small proportion of patients can have secondary lymphoma tumors, but this is still controversial because it can be also due to the chemotherapy that the patient received before. So for sure is safer than chemotherapy. And for us, it's very, I'm very anxious to see a therapy approved for solid tumors because these are more difficult to treat, and all the approved therapies are for hematological. And I think there are some reports, no? Some clinical reports seeing nice results, but not long term. For instance, for this DIPG or ATRT or DMG that have a 6-month average survival, with the first clinical trials seeing an improvement of 6 months. So they are reaching from 6 months to 1 year. This is not a lot, but it's 2 times more than we had before. And now the challenge is to obtain a CAR T that works better, no, that has more, that has less exhaustion, and that goes for more time, etc. So a lot of work to do, but with a high potential, I think.
Okay. Well, I see no more questions appearing in the chat. Is there anything else from your side that you want to add, Ciprian or Jordi?
Just thank you for the invitation.
Yes. Thank you very much.
Well, okay. I also thank you both again, both of you speakers, and I also thank all the participants, everyone who watched us today. I will finish this session, and the recording will be made available soon. Yeah, so everyone who didn't have the opportunity to watch it can still watch it later. So thanks again, and have a nice day. Bye-bye.
Bye.
Bye.